So good day, ma'am. I am your student in this subject, AG 206 biochemistry. So my name is Sheila Tibaldumar. I live in San Isidro, Pamplona, Negros Oriental. So I am presenting this video, lesson 6. So question number 1. In your own words, how is a secondary, tertiary, and quaternary is protein structure formed? Discuss briefly. So the secondary structure is from the polypeptide backbone, forms a repeating helical structure that is stabilized by the hydrogen bonds between a carbonyl oxygen and an amine hydrogen, and these hydrogen bonds occurs at regular interval of one hydrogen bond. And also the amino acid and cause the polypeptide backbone to form an alpha helix. The tertiary structure is form of a protein, refers to the overall three-dimensional arrangement of its polypeptide chain in space. It is generally stabilized by outside polar hydrophilic hydrogen and ionic bond. So interaction and internal hydrophobic interactions between nonpolar amino acid side chains. Internal hydrophobic and interactions between nonpolar amino acid aside the chains. The quaternary protein structure formed of consisting of two or more identical or different polypeptide chains, or the subunits. These proteins are called oligomers because they have two or more subunits. The quaternary structure describes the manner in which subunits are arranged in the native proteins. Quaternary structure protein is form of association of the several protein chains or the subunits into a closely packed arrangement. So the subunits are held together by hydrogen bonds and between nonpolar side chains. Question number 2. What is the difference between supersecondary protein structure and protein motifs? The supersecondary structure is usually composed of two secondary structures linked together by a turn and includes helix turn helix, helix loop helix, E alpha corners of B beta corners, and B hairpin beta. Protein motifs are more complex structures created from secondary and supersecondary structural components that are repeated modalities visualized in many protein structures. Question number 3. What are the characteristics of a fibrous protein, globular protein, membrane protein, disorder protein, and the molecular chaperones protein? Fibrous proteins are characterized by elongated protein structures of the protein often aggregate into filaments or bundle, forming the structural scaffolds in biological systems. Within animals, the two most abundant fibrous protein families are alpha-keratin and collagen. The globular proteins or spiroproteins are spherical proteins and are one of the common proteins types. Globular proteins are somewhat water-soluble, forming colloids in water, unlike the fibrous or membrane proteins. The peripheral membrane proteins, which are attached temporarily to the membrane via integral protein or the lipid via layer. So the disordered proteins and intrinsically disordered proteins, or IDP, is a protein that lacks a fixed or ordered three-dimensional structure, or IDP cover a spectrum of states from fully unstructured to partially structured and include random calls pre-molting, globules, and large multi-domain protein connected by flexible linkers. So the molecular chaperones proteins are the chaperones exist in all cellular compartments and interact with the polypeptide chains in order to allow the native three-dimensional conformation of the protein to form. The chaperones themselves are not included in the final structure of the proteins that are assisting. Question number 4. What is a practical example of protein denaturation in our daily life? So the example of protein denaturation in our daily life. When food is cooked, some of its protein become denatured. This is why boiled egg become hard and cooked meat becomes firm. Of the denaturing in proteins come from egg whites, which are typically largely egg albumins in water fresh from the egg. Egg whites are transparent and liquids.